It's a really great pleasure to welcome all of you here today for this webinar session on science policy for biodiversity research. The sessions today and on April 24th have been organized as a collaboration with the Global Genome Biodiversity Network, so the GGBN, and the European Reference Genome Atlas, or ERGA, with the support of the Digital Sequence Information DSI network and the Earth BioGenome Project, the EBP. My name is Robert Waterhouse, based here at the SIB Swiss Institute of Bioinformatics, and representing ERGA as the current chair of our initiative that now counts more than 1,000 members. So the ERGA community is a community of scientists that are aiming to work together to try to coordinate and facilitate the generation of reference quality genome assemblies for the entirety of European eukaryotic biodiversity. This is clearly a very ambitious and complex endeavor that will take a huge concerted effort of a great deal of people. And as we've been getting started in this endeavor, it is really becoming more and more clear that policy for biodiversity research is a particularly complex issue, where we have many ERGA members who are seeking guidance and help to make sure that they are following the best practices for ethical and legal sampling of species across Europe. So on behalf of all ERGA members, we are very much looking forward to learning from the experts today during these two webinar sessions on science policy for biodiversity research. And just before I hand over to Jonas Astron, who will be representing GGBN today, we're going to first launch a very quick poll here on Zoom and ask those of you who wish to receive a certificate of attendance for today to please enter your first and last names, as this will allow us to register those present today on the call and send out the certificates of attendance later this week. So hopefully we can launch the poll now, and then it's fairly simple. If you've seen how to do this on Zoom before, you just enter your first and last names and hit submit. So then we have a record of attendance for today. So while you're doing that, I will hand over to Jonas, who will introduce the GGBN.
Thank you, Rob. Good morning, everybody. Also from my side, I'm also very happy that this seminar is taking place and in conjunction with various important players in the field. I am the curator of the biobank at LIB, Leibniz Institute for the Analysis of Biodiversity Change at Museum König in Bonn, Germany. But my role here today is that of the chair of the executive committee of GGBN. GGBN, for those of you who haven't heard of it, is the Global Genome Biodiversity Network. A bit clunky name. What it really hides is GGBN is the network of biodiversity biobanks. So we have lots of biobanking happening in the human realm, but GGBN is really the platform to bring together the institutes that work on biodiversity molecular samples, archiving them in biobanks or molecular collections or whatever you want to call those. There's lots of names out there for the same principle. And in GGBN, we have about over 110 member institutes in almost 40 countries worldwide that come together to make their collections visible through a platform, GGBN. Thank you very much, Rob, for putting the link in there. To GGBN. GGBN offers a platform to have people searching for molecular data in collections, in biobank collections, show the data online through that collection. Also, it brings together the biobanks to have a platform to talk together, and GGBN also focuses on standards and best practices. So these are basically the main lines of activity within GGBN. And we have regular conferences, biannual conferences. The next one is going to happen in Cape Town in October next year. The last one just was in Aguascalientes. So yeah, that's GGBN in a nutshell, and I won't go further into any further details, and let's continue to hear about who is our speaker today. I think that's for you, Luisa, to introduce. Thank you, everybody. Bye.
Thank you, Rob and Jonas. So I will quickly introduce Scarlett. Dr. Scarlett Sett has a research background in biological oceanography and has been working in the field of ABS, access and benefit sharing, since 2017. For the past 6 years, she has been working on the practical implications of ABS and the Nagoya Protocol on academic research. She was hired as the first ABS compliance officer in Germany at Kiel University and developed a strategy for the university to foster compliance to the Nagoya Protocol and to ensure researchers understand their responsibilities and obligations in regards to the EU ABS regulation. Scarlett currently works in the context of pathogens and ABS at CSIRO in the Australian Centre for Disease Preparedness. So yeah, that was the short bio, and now I think we're ready to start. Thank you again, Scarlett, for accepting to give the talk.
Thank you, Lisa. Thanks for the invitation. I hope everybody can hear me well. Yes, I'm in Australia. It looks light outside. It's 5 p.m. So if I say something wrong, it's because it's getting to the end of my working day. But I hope I will not say any nonsense. Yeah. So the first things that I like, one of the first things that I like to do is get to know you a little bit better. So to know who is in the audience, because I cannot see you. So to create a little bit, maybe in some areas I can target the information a little bit better. So I'm going to be using this platform. It's called menti.com. So you can either put that on your browser, or you can scan the QR code with your phones, and then you can then use that code that is there to access the poll that I'm going to be running. And so I just want to know if you know why you're here. And this is just to test also the program to see if people can access it. Like I said, at the top of the screen, you can always see Mentimeter. Okay, good. That's good. I'm happy. I'm happy that people. Oh, still, okay. Still unsure. I would be worried if there would be people in the middle, but the unsure I can understand. Okay, you have 35, 36. Okay, so that, and we had like 49 people so far, right? That's, oh no, 81 already. Okay, so, okay. Just give it a little bit. Okay, good. I'm glad. I know it's Monday morning, so everybody's starting their week already with a heavy talk. So thank you for being here. Okay, I'm gonna go. Okay, just a few more seconds, and I'm gonna go to the second question. And the second question before you vote is, I want you to put it's like a word cloud, so you're gonna have to type. Please just use the normal names, don't like in English names of the country. So then if people say Germany, then it would, like, if you put Germany and someone else put Germany, then the word is going to get bigger in the word map. So, and I need to know which country you're working. So it's not about your nationality, it's about in which country you're working, or, you know, if you have two institutions, in which institution is the one where you carry like your actual laboratory work, so to say. Okay, Germany is a big contender. Let's see. Okay, Germany's still in the middle. Wow. I wonder if there's a lot of Germans or the system is just rigged. Okay. That's pretty cool. Also Switzerland. Okay, there was a typo, but I'll take it. Anyways, okay, that's really good. And then, okay, a couple of more people logging in. Okay. The next question that I have is about where is the country of origin of the material that you are using. So let's say you're working in Germany, but the material that you use for your research comes from Costa Rica, or I don't know, South Africa or something. So if you have more than one country that you material that you work with, then put more than one country, but this is about the country of origin of the material that you use for your research. Okay, so this is interesting, and this will be very This will be very nice at the end to compare where people are working and where they're using the material for, like the physical material. So. Worldwide, I mean, yeah, I know, but I mean, it's quite broad. I don't think you're using 196 countries, but I mean, nice try. Let's see, everywhere. Okay. And this is the last question before I get started. It's about your level of knowledge about this topic. So how much do you know about ABS and the Nagoya Protocol? So are you a beginner? So really you just know that there's something called the Nagoya Protocol, but you don't know much more. Somewhere like in the intermediate, so you know a little bit, but you still have a lot of questions, or you're an expert and you just want to check whether I'm talking some nonsense today. Let's see, 4 people, okay. Maybe those 4 people are also the one from my team at the DSM Sett, so that would be okay. Okay, but this is usually what I would expect. I have somewhere between beginner and intermediate. The majority of people are between beginner and intermediate. So I hope that for the beginner section, you will leave this also for the intermediate. Usually if the intermediate people are here, because they still have a lot of questions. So I'm hoping that those questions will be answered. And for the beginners, I hope they will leave today after these 2 hours with a better understanding and whether this is something that will affect their research. So this is the outline of my presentation. So I will start with putting it into context. So why is ABS important? What does it mean for you in practice? So what are the practical implications of this framework? And why is it important that you comply with these regulations, right? Are there consequences? And if there are consequences, what those consequences are? And at the end, we will have a Q&A session. I think someone mentioned it, right? But just the first part of this presentation will be recorded. The Q&A session will not be recorded. So feel free to ask any questions if there are still some remaining ones at the end of my presentation. I will not be taking questions within the presentation, but at the end, so you can put them on the chat or you can just write them down and then we'll address them during the Q&A session. Yes, pay attention because you're going to be tested at the end of my presentation. So we're going to use Mentimeter again just to test whether you got everything, and then we can see how you did, if you were paying attention or you were awake. So putting it into context, so why is ABS important? And again, ABS stands for Access and Benefit Sharing. And I think if you are from the GGBN or the ERGA community, then you do have quite a close working relationship with biodiversity. So this graph might look familiar to you. So I won't go into detail, but it basically just shows that there has been a very strong loss of biodiversity since the 1970s, and that is quite strong in the tropical and subtropical areas where you find these hotspots for biodiversity. So biodiversity loss is a big concern and then links quite nicely to what we want to cover today in the workshop. And for me to talk about access and benefit sharing, or ABS, and the Nagoya Protocol, I have to start from the beginning, and I need to briefly say something about the Convention on Biological Diversity, or the CBD. So the CBD was created as a recognition that biological diversity is a global asset to present and future generations, and with it, the threat to species and ecosystems has never been so great as it presently is. So currently it has 196 parties, with the US as an observer party, and it has 3 main objectives. So the first one is the conservation of biodiversity, so biological diversity. The second one is the sustainable use of its components, and the third is the fair and equitable sharing of benefits arising from the utilization of those components. And it is on this last point that the Nagoya Protocol then takes off. And this last objective was part of the deal in the political negotiations to engage the developing nations into increasing their biodiversity commitments, which then later on led to the development of the legally binding framework, where they would also be able to generate benefits if someone used the resources. And the Nagoya Protocol is a supplement agreement to the CBD, and it aims at providing transparency and clarity on how to comply with access and benefit sharing obligations arising from the utilization of genetic resources. This is important for you. So entered into force on the 12th of October of 2014, and it currently has 141 parties. Unfortunately, 10 years later, there's a large proportion of scientists that are still unaware that this exists, what it entails, and how it affects research activities. But its goal is to help establish conditions for the access to genetic resources and to ensure that benefit sharing occurs once that genetic resource leaves the providing country. So in a nutshell, what you need to know about the Nagoya Protocol is that it covers genetic material that within national jurisdiction. So in land, usually it's, the borders are pretty clear. That extends up to the 200 nautical miles at sea, and it covers all genetic resources. So that's non-human material that contains DNA or RNA, whether it's dead or alive, and research and development conducted on the genetic and or biochemical composition of those resources. So it also includes research and development that is carried on derivatives. So that's, for example, proteins, enzymes, secondary metabolites. It covers materials both in situ or ex situ and affects both commercial and non-commercial research alike. So the next slides are quite tedious, so just bear with me because I just have to walk you through the general basics. So the Nagoya Protocol establishes international rules and obligations for its parties to take measures in relation to the access to genetic resources, benefit sharing, and user compliance measures. So this is known as the ABCs of ABS. So these are the 3 pillars of the protocol. So access obligations deal with the permits you might be required to obtain to access or collect the genetic material from that country. This is usually in the form of a prior informed consent permit, which is granted by the providing country as a prerequisite to access the genetic material. When I talk about providing countries and them giving you permits, I'm talking about like the country will designate an authority that will be able to represent that country. Usually it's the Ministry of Environment. It can be also other kind of government offices, but it is an official authority that the country has designated to have that responsibility to be able to give you permits to access that material. Then you have the benefit sharing obligations. There are measures to provide for the fair and equitable sharing of benefits arising from the utilization of those genetic resources. And these are based on mutually agreed terms, so MATs, which are contractual obligations between the provider country and the user. When I'm talking about the user, I'm talking about you, the researchers that will use that material, right? So do not confuse the MAT with an MTA. The MTA is a material transfer agreement, and probably if you work at the university and receive material from abroad or from other institutions or for companies, you've heard about this term. It's not the same. So MAT, mutually agreed terms, MTA, material transfer agreement. And benefit sharing agreements can be monetary and non-monetary. And I list here a few examples of what this could be. For example, monetary benefits could be access fees or payment to royalties, research funding. So when you give like actual money in exchange for something else. Then you have the non-monetary benefit sharing, which is more in the like education and training. If you have collaboration in scientific research programs with that country and an institution in country, institutional capacity building. So for most of us, I assume in this workshop, we will be in the non-commercial academic sector, and most likely the benefit sharing that we will do will be non-monetary. And then you have the C, which is for compliance, the compliance obligations, and these are specific laws, administrative tools, or policy instruments that the Nagoya Protocol requires its parties to establish for the users of the genetic resources to ensure that they comply with any rules established within the jurisdiction of that providing country. So these are the 3 basic rules of the Nagoya Protocol. Now, this is where it starts to get more complicated. So access and benefit sharing obligations will be established and negotiated between the provider country and the user of the genetic resources. Again, the providing country, that will be the authority the country had designated to represent them. Again, it could be a ministry, but it will be probably a government office. And ABS access and benefit sharing are regulated at a national level, that is under laws of the country that provides those resources. So the compliance obligations are regulated and implemented at an EU level by means of the EU ABS regulation 511/2014. And therefore, it's very important for all of you working at institutions in the EU. This means that researchers working for institutions in any of the EU member states are required to show documentation of the legality of access to their samples and that they are complying with any with rules on ABS established by the country that provided those resources. But how does this practically affect your work, or what do you need to pay attention in order to be compliant with the EU ABS regulation? So you see, I don't know if you've noticed, we've come from the very broad ABS term to the Nagoya Protocol, and now we're going into the EU ABS regulation. So to determine if you are within the scope of the EU ABS regulation, you need to fulfill certain requirements. So in very simple terms, although it's not a simple thing, although in very simple terms, there's 4 questions that you need to answer, and we will go through each of these questions. So a very important point is that you have to answer yes to all of them to be within the scope of the regulation. Otherwise, if you answer no to any one of them, then you might not have obligations with the EU ABS regulation. However, and this is why, you know, I don't like to say this, but however, it could be that you do not have EU obligations under the EU ABS regulation, but you might still have obligations with the country that provided you those resources. If that sounds confusing, I'm going to give you some examples later on of what that case could look like. So the first question is about what kind of material you are using. So this is called the material scope. And we will use I'm going to be using the definitions that are provided on the CBD and the Nagoya Protocol frameworks with the EU, which the EU ABS regulation also takes. And a genetic resource is defined as genetic material of actual or potential value. So basically all living organisms containing DNA, like plants, animals, microbes, carry genetic material that could be potentially useful to humans. So we just don't know it yet. So everything that you use could potentially carry super important information, or does carry super important information. And any genetic material is any material of plant, animal, microbial, or non-human origin containing functional units of heredity, so genes. And any material that contains DNA or RNA, whether it's dead or alive, would be considered genetic material. So these resources can be found in the wild, domesticated, or cultivated, but they are sourced from environments in which they occur naturally, so in situ, or from human-made collections, such as botanical gardens, or gene banks, or microbial culture collections, so ex situ. So some exceptions to this is human DNA, but this does not include human pathogens and the microbiome. So if you are studying human pathogens in isolation to their host, then this would be then within the scope of the EU ABS regulation. There are also some other material, like certain plant varieties that are covered under the International Plant Treaty on Genetic Resources for Food and Agriculture, or influenza strains that are covered under the Pandemic Influenza Preparedness Framework. So as long as the genetic material is covered by another instrument, it will not be covered by the EU ABS regulation. However, just keep in mind that, for example, if you get a plant that is covered under the plant treaty and you're using it for something else than for food and agriculture, so you're using, let's say you found this plant that you think that you can use it for cancer treatment, then this would again fall under the scope of the EU ABS regulation and the Nagoya Protocol, because it's not under the scope of what the plant treaty was made for, which is for food and agriculture, right? So just keep these things in mind. Don't say, oh, because this is from the plant treaty, then I don't have to do anything. So of course it's under the plant treaty if you're using it under the conditions of the plant treaty. If you're using it for something else, then it falls again under the scope of the EU ABS regulation and the Nagoya Protocol. The second question is about what you are using this material for. So, and the utilization of genetic resources, again, we're taking the definitions from the frameworks, is to conduct research and or development on the genetic and or biochemical composition of genetic resources. Again, this also includes research and development on derivatives, so proteins, enzymes, secondary metabolites, although these molecules themselves do not have units of heredity. And misinterpretations of the definition of genetic resources and the utilization thereof has been one of the greatest misconceptions of the scope of the Nagoya Protocol, and that it affects both commercial and non-commercial research alike. And I'm providing here some examples. These are extracted from the EU guidance document of research and development that fall within the scope of the EU ABS regulation. For example, research on genetic resource that will lead to the isolation of a biochemical compound that is used in an ingredient for a product, whether it's active or not. A breeding program to create a new plant or organism variety, genetic modifications of an organism, and again, research and development on derivatives. And then some examples of research and development that do not fall within the scope. So research that exclusively deals with taxonomic identification of organisms, which might also include analysis, does not fall into the scope of the regulation. However, this can be regulated under national law of the providing country. Genetic resources that are used as testing or reference tools. This includes animals used, for example, animals or whatever used for testing medical reactions, so yeast in the brewing process. So it does not fall into the scope of the EU ABS regulation when the genetic resource is not in question, is not the research question, but just means to get to something else. We usually have a lot of questions about plasmids, for example, and this is one of the great examples. If plasmid, usually you just buy the plasmids as means or vectors to get to something else. So in that regard, you would be out of the scope of the EU ABS regulation. However, if you're interested in the plasmid itself and looking at the genetic composition and biochemical, then that would be then under the scope of the regulation. So handling and storing of biological material for descriptive purposes, for example, this is also out of the scope. If you just want to compare something in the size for something that was 50 years ago, this you wouldn't need to do, it wouldn't be under ABS. And studies that also deal with purely descriptive purposes. So for example, if you put a tracker on a lion and you want to see how it moves around, if you're not looking into the genetic and biochemical composition, then this would be out of the scope. The next question, so this is the third question, is relatively easy. It is about when you access the material. So since the protocol is not retroactive, what is important for you is that the material was accessed on or after the 12th of October of 2014, which is the date in which the Nagoya Protocol comes into force. Remember, however, that some countries have access and benefit sharing rules that were established before the entry into force of the Nagoya Protocol, and this would then be applicable. So you might still need to collect special national permits, although they're not under the Nagoya Protocol. So there are also, just to add more confusion to the whole thing, there are also some countries that consider access when you start using the material for research and not when it was initially collected. So for example, this is France. It defines access when you start using the material. So it could be that you have a plant from France that was accessed in the 1920s and you're just going to start using it now. So under their interpretation, you would have to get access permits now because you are just starting to use the material now in 2024, although the material was collected in 1920. And the last question is about where the material is coming from. So is the provider country party to the Nagoya Protocol, and does this country have national ABS regulation in place? And you can find if the country is party to the Nagoya Protocol, if you look into the ABS Clearinghouse website, here is the address. And the ABS Clearinghouse website was supposed to play a key role for the implementation of the Nagoya Protocol, and it was created to provide a platform to exchange information on ABS regulations between the user and the provider of the genetic resources, and in particularly to provide the users access to information made available by each providing country regarding the procedures for access, and that the providing country could also do some sort of monitoring of utilization. So each party was required to publish their respective information on national focal points and competent national authorities, and also if they had any ABS measures in place, that they would be uploaded here. So if you want to know whether that country have ABS measures in place, basically each country can decide if and how they regulate their resources as long as they comply with other relevant laws. So for example, this is a map of the EU. Within the EU, you have Spain, France, Bulgaria, Malta, and Greece. They have their own ABS regulation, and processes that you have to follow when you want to access their material. While Germany and the Netherlands, Denmark, you can go and freely access and utilize genetic resources without any access permits. And it doesn't seem that this will change in the future for those countries. So the red countries are the ones that we are for sure, we know that those countries required ABS process. The yellow ones is like sometimes they require depending on the condition. So this is just to give you an idea that even within the EU, there's a broad range of interpretation on how they decide to regulate their resources. So you might find this information, whether there's an ABS process or not in your desired country, in the ABS Clearinghouse website, but unfortunately it's not always complete. So don't trust blindly. If the information is not clear and you have a feeling or you've heard rumors or an indication that they might have ABS rules, please contact the national focal point of the provider country and ask them for further information. So these people listed there should be the ones that will be able to answer your questions. And this is super important for me to mention. Again, do not rely on your in-country collaborator partners, as they might also not be aware about their national laws, but contact the national authorities. And we've had a lot of issues, for example, with Spain and France, that they say, well, I'm a Spanish researcher working in a Spanish institution. I don't need to do anything. And we're like, sorry, but you do actually. You do have to go through a process if you fulfill the requirements, right? So sometimes the laws that apply to national researchers will not apply to you. So it's not like it's not like your in-country collaborators are giving you wrong information. It's just that the rules that apply to you might be different than the ones that apply to them. So don't trust your in-country partners. Directly contact the national focal point and ask them for further guidance. So if you answer yes to all of these questions, then your research, congratulations, your research is within the scope of the EU ABS regulation, and you will be expected to follow user obligations as stated in Article 4 of the ABS regulation. Now, people start to freak out when I tell them that you are within the scope of the EU ABS regulation. If you got to this point, it means that you already did all the work. So what you're going to have to do here will be minimal. So there will be some steps, but it's minimal, so you don't have to worry. It's not extra stuff that you have to do. If you got to this point and you are under the scope of the EU ABS regulation, then you're basically set to just do this next step. So this mainly deals with the exercise of a due diligence declaration, so regarding the legality of access and benefit sharing obligation, and this can be done through the Declare portal of the EU. If you're in the EU, you should ask your institution whether they already have an institutional account. Sometimes that makes it easier because it means that it has already been registered with the national authorities that will check whether your permits are correct. And the declaration enables the user of the genetic resource to submit the due diligence declaration to the relevant competent national authorities responsible for implementation. So for each country, there will be a national agency that checks if the documents are correct. That's to say, if you work in Germany and you submit a due diligence declaration, that declaration will go to the German authorities. And the German authorities then go through the documents, check with the provider country, make sure that everything is correct. And then they will extend a checkpoint communique and put it into the ABS Clearinghouse website. That means you're good to go. And of course, if it's another country, then other countries' authorities will get your declaration. So it is important that as a user of genetic resources, you always keep these documents at hand. So that's also part of your user obligations, that if you transfer this material to a third party, if you are allowed by the MAT, you would also transfer this information along to the next user and vice versa. So the fact that there's no ABS regulation in place in some countries, which are now party to the Nagoya Protocol, does not mean that it will remain like this. So for example, within the EU, there are some countries that are just starting to develop their own national laws. And therefore you should always keep in the record when you access the material, because this will make a big difference if someone comes and checks you and they say, well, I accessed the material in January of 2017. This is a hypothetical thing, but I accessed the material in January of 2017, and then the legislation started from October of 2017. So you can still prove that when you access that material, there was nothing that you needed to do. And this will save you a lot of trouble in the future. I think you should anyways keep this information. I think good scientific practice is just that you keep this information, but just this is just an additional ABS tick box that you can say, okay, let's keep always the information when it was collected and where it was collected. Yeah, also this also goes if you're getting material not yourself from that country, but if someone else is giving you the material, whether they're giving you the relevant information. So again, I sound like a broken record, but you need to start with when the material was collected and where it was collected. And from there, you can like work out whether you need anything or not. So it is your yeah, they provide you with the required documentation. Am I actually permitted to do my research? So if the material was accessed through an ABS permit, you should get those permits, like a copy of it. You don't need the original, you need a copy, and you need to check whether actually your collaborator, can they actually transfer to third parties? So all the permits that I've worked with Kiel University, all of them had a specific clause that said a third-party transfer was prohibited. So this is just like a normal clause for them. So most likely, in most cases, this will not be allowed. So it's your responsibility to check those permits from your collaborators to see whether actually they are allowed to transfer the material to you. If they're not allowed to transfer the material, you shouldn't accept it. Also, are you permitted to do the type of research that you want to do? Because if the permits were given to do ABC research and you want to do something completely crazy differently, this would also not be allowed. You would have to go back and renegotiate those permits to fit the utilization that you want to give them. Do benefit sharing obligations continue to apply? Remember that the MATs, the mutually agreed terms, are a contractual obligation between the country that gave you those resources and the user. And they could have said, okay, benefit sharing will be that you are going to come once a year to give capacity building, that you're going to give a workshop to our in-country scientists every 6 months, I don't know, whatever. And then you have to ask, do these benefit sharing obligations also continue to apply to me, although I was not the person that was originally getting the permit? Are there any restrictions or conditions on use for the material, for example, for non-commercial use? And if any obligations will be transferred to other parties. So this is, again, important. Material transfer agreements, or MTAs, between institutions alone are not sufficient to fulfill your information obligations. Again, an MTA is something between institution and institution. What you need for your ABS agreement is an provider, country, and user. Also, exceptions for local scientists don't apply if the material leaves the country. So this is also like some of the things it could be that your in-country collaborator said, no, I didn't have to do anything. And it could be because they are an in-country scientist and they didn't need to do anything, but this might be different to you if you are coming from abroad and then also taking the material outside of that. So it is also your risk. So you and your institution need to make a risk assessment if it's worth it to work with the material that you can approve it was legally acquired. So in the EU ABS regulation, there's no prohibition for you to use material that you don't know where it's coming from, but you just have to make a risk analysis whether it's worth it to do it. And if that country at some point finds out that you're using material from them, whether those risks are enough to take. And users are obliged to retain information on access and benefit sharing for a period of 20 years after the end of utilization. And before people start to freak out, this again only refers to documentation, so not the physical sample. And it also means, for example, if you obtain the material in 2020 and you use it for 20 years, so the obligation for storage of documentation would start in 2040, so at the end of utilization, and then the 20 years start to count. By now, you're probably wondering, oh my God, this is so much. Do I actually have to pay attention to this? Do I want to even do this? Well, friends, I'm sorry to tell you, there are consequences if you're found non-compliant. So again, because most of my work has been in Germany, there are no criminal sanctions, so you are not going to go to jail, but there are regulatory offenses that can amount up to 50,000 euros. So more detrimental to institutions is that genetic material can be confiscated and research projects can be stopped. So if you got a grant and they find that you have been non-compliant, they can cancel your research project. So in Germany, again, fines have already been given to an institution that could not prove the legality of access, and the material and research results were embargoed, as well as the theses that were of the people that were writing the theses that used the materials. Those were embargoed and they couldn't go on with their studies. So in other countries like India and Brazil, researchers have already gone to jail for not complying with ABS permitting. And theoretically, you could be then sanctioned twice. One, in your own country for not complying with, if you're working within the EU, in your own country, if you're not complying with the EU ABS regulation, and two, with the country in which the resources come from. So some of the non-regulatory consequences are loss of reputation and trust with the provider countries, and that no one wants to be labeled a biopirate. And these last pieces in science since 2019 already show that some journals might start to develop their compliance checks to ensure material being published has been legally acquired. So journals will not have the authority to check whether the permit is actually right or not, but they can still ask for the legality of access. And some countries might even go as far as banning scientists from entering their countries and blacklisting them and their institutions. So you don't want to risk this. I mean, we also don't know how this blacklists work. I mean, is it a whole institution? So even if you leave after 2-3 years, will this institution still be blacklisted? And then you will prevent other researchers to go to that country and do research there. Okay, so now that I have overwhelmed you with all this information, let's see. Let's go into the quiz before I go into my take-home messages. So again, you can use you can scan the QR code, or you can use the menti.com, and again, the same number. You can always look at it at the top. So first question, are you awake? And can you answer all the questions correctly? Okay, well, that one person, probably the one that also said didn't know. This question is about, were there ABS laws before the entry into force of the Nagoya Protocol on the 12th of October of 2014? Yes or no? Let's see. What do you guys think? Don't be shy. Okay, the right answer is yes. So there are countries that have had ABS laws preceding those of the Nagoya Protocol: South Africa, Costa Rica, Colombia. There's a lot of countries that have ABS laws way before the Nagoya Protocol on the 12th of October of 2014. And those are the ones that you have to keep in mind if you're getting material from some country that might already have ABS laws before the Nagoya Protocol came into force. The second question is, do all parties to the Nagoya Protocol regulate access to their genetic resources? Is this true or false? Which in this case it is. That's false. So countries can decide if and how they regulate access to their genetic resources. So they don't have to do it. Again, for example, Germany, Denmark, the Netherlands, they don't regulate access to their genetic resources. So everybody can decide if and how they do it. They can also say we regulate some stuff and we don't regulate other things. Portugal, for example, is a good example. Mainland Portugal, like, resources are not regulated unless they come from natural reserves or protected areas. But Madeira, the Azores, and other islands, they do regulate access to the resources. So they can decide how they regulate it and if they do some sections and some other sections not. So this is a case. So I'm conducting research on material from a country that is not party to the Nagoya Protocol. I do not have ABS obligations. Is this true or is it false? But the correct answer is false. So the fact that a country is not party to the Nagoya Protocol does not mean that that country does not have ABS laws in place. So you will always have to look whether the country have ABS laws that you would still need to fulfill if you want to access material from their from them. Okay, this is the next question. My research will not produce a commercial product and I will not generate any profits. I have no benefits to share, and ABS, the Nagoya Protocol, do not apply to me. Is this true or false? Okay, so it's good. The majority, the right answer is false. That it doesn't matter that you're not doing commercial research, and it also doesn't matter if you will not produce money. So benefit sharing can also be non-monetary. So you can still be contributing with non-monetary benefit sharing. And the next question is, I do genetic research on a tropical fish I bought at a pet shop. I have no ABS obligations. I'm good to go. Is this true or false? This is a tricky question because I mean we put these questions there because there's a lot of people that come with this kind of scenarios. So we think that maybe it will apply to some of the people that are in the audience. This also applies, for example, if you bought something at the supermarket, you know, like you bought it in a store and they were allowed to sell it. So why wouldn't you be able to use it for research and development, right? For your research project. So the correct answer is false. So even if you buy something from a store, so for example, in this case, you bought a fish on a pet shop, the trade of the fish into that pet shop was with the intention to have it in an aquarium for decoration or whatever, not to do research and development. So if you want to do research in this tropical fish, you would need to know where it is coming from and then get permits from the country. If they require permits, contact the national authority from the country where the fish come from and see whether you need to have any ABS permits. So the same thing if you buy something in the supermarket, it came into the supermarket for the intent to be sold as food and consumption, not for research, and therefore there's a change of intent. And therefore you would then need to go back to the providing country and ask if you need to do anything to utilize that material for research. The next question is, I cannot start my research project until my ABS permits have been granted. Is this true or false? I think there's only 2 more questions, so bear with me. So the correct answer is true. You shouldn't start with your research project until your ABS permits have been granted. Remember that if you have to go through an ABS process, you need 2 things: the PIC, the prior informed consent permit, and the mutually agreed terms. Sometimes this could be just 1 document, but one will give you the permission to access the material. The other one, what is it that you need to give in exchange for the utilization of that material. So those 2, you have to sign those permits before you start actually using it, because you cannot start using it without knowing what is it that you have to give back. Okay. A colleague in Spain collected biological material in France, and he transferred the material to me via an MTA. Let's say me or the user is in Germany. I have no more responsibilities. The MTA is enough. And I trust my colleague in Spain. That is correct. So the right answer is false. I don't know. I don't think I clicked. The right answer is false. So an MTA is not enough to fulfill your obligations. So then again, an MTA is just between institution and institution. It doesn't mean that the country gave you the permission to work with that material. And from this question, we're going to go like to the follow-up question. So then the follow-up question would be that this same colleague in Spain that collected the biological material in France, you working in Germany, it gives you the MTA. They say, well, I need to know more. If you ask, which is the country do I need to contact regarding ABS laws for the material that you want to work with? Is it Spain? Is it France? Is it Germany? Or both? Or all of them? This is the last question, guys, I promise. And then that's it. Then we can move to the take-home messages. Yeah, so the right answer is France. So remember, your colleague in Spain collected the biological material in France. So the providing country is France. You would need to contact the French authorities to see if there's anything that you need to do in order to use that material for research. Yeah. So to wrap up, what are your responsibilities as users of biological material? So first, do your homework. It is your job to inform yourself, and if you have any obligations with the providing countries, don't blindly trust your in-country partners, as they might also be unaware of their own rules. So it is a lot of information, and I'm sorry about that, but it's just the way that it is. So break it down into manageable steps and make a checklist to make sure you are not forgetting anything. So know what questions to ask and where to look for trustworthy information. Again, you can use the ABS Clearinghouse website. We also have a wonderful project in the DSM Sett called the German Nagoya Protocol Hub. We've also developed a checklist for users. So I hope that one of my colleagues can share the link with you. So use those resources to guide you through the process. Ask for help if you are unsure. So you can ask your institution whether you have a compliance officer that can support you through this journey. In Germany, there are a lot of institutions that have employed compliance officers just to deal with ABS. Again, in the German Nagoya Protocol Hub website, you can find a list of German institutions that have compliance officers. So refer to them if you have questions. Otherwise, we also have a help desk contact form in the German Nagoya Protocol Hub website. If your case is really complex and you have no one else to help you with, you can also then send an inquiry through them, and one of my colleagues will be able to help you. And this last step is also very important. Check with your supervisor and legal department before you enter into any legal obligations. So these mutually agreed terms and contracts are legal documents, and you need legal advice before agreeing to those terms. So at least at the university, only professors or someone with a permanent position were able to sign these documents, and they would always do it with support from the legal department, because if you get in trouble, especially because it's most likely abroad, then you need to make sure that the laws would that you will be protected somehow if something goes terribly wrong. And some take-home messages. Of course, these are very simplified because these are very complex topics, but ABS is complex and goes beyond the Nagoya Protocol. So there are countries that are not party to the Nagoya Protocol, again, with existing and very strict ABS laws. So make sure you comply with them. Keep in mind that you might still have national obligations that go either above the EU ABS regulation or the Nagoya Protocol, and you still have to comply with this. You have to respect them. And again, I like to use this circle because you have, like the bigger framework, so ABS, and within ABS, you have the Nagoya Protocol, and within the Nagoya Protocol, you have the EU ABS regulation. They're not mutually exclusive, right? So you can still be within the EU ABS. You cannot be within the EU ABS regulation, but that does not automatically mean you do not have ABS obligations. Also, and I didn't add this here to the graph because I didn't want to just add more confusion to the whole thing, but ABS also goes beyond the CBD and the Nagoya Protocol. So there are also other frameworks that deal with ABS or have ABS systems in place. If you work with marine resources, you might now be aware of the BBNJ treaty. So there might also, this could be like an extra circle there, or the pandemic influenza preparedness. That's also another circle in the blue circle, or the new WHO pandemic treaty. That would be an extra circle that comes on top of the ABS, outside of the Nagoya Protocol and outside of the EU ABS regulation. So it is complex, and there's a lot of also other things that have ABS components outside of the CBD and the Nagoya Protocol. The system is not perfect, so we are all aware of this, and we all hated it at some point, and now we're just like getting to the point you're like, okay, this is just acceptance, we just have to deal with it, there's nothing we can do. Unfortunately, it's not a transient topic, so it is an established legal framework that took years in the making and that we have to comply with. So if we do not comply with our obligations, there will be consequences, and they will be severe, especially as time moves on. The more countries will develop their own laws, and they will be able to monitor where the material is going for, is being used. Take it seriously. You do not want to risk your reputation and your research future if you are found non-compliant. Again, you might not only be putting yourself in a difficult position, but other future potential researchers that want to work in certain countries. So be careful about that. It's not like, oh, I don't care, let's see what happens. I'm not going to get caught. Just be very aware of that, right? Do not underestimate how important it is to have a strategy for your project. So inform yourself in time, and if you need permits, start as early as possible. At the university, we would start 6 to 9 months early just to get the process going. Most of the ABS processes don't have any cost. It's just communication, right? And negotiation of what is it going to be in the permits. So start as early as possible. Even if you don't have the money for the grant yet, start early. If the money doesn't come, then like at least you would have been able to start when the money comes and not wait until the last minute. Sometimes it could take 2 months, sometimes it could take 2 years. So just keep that in mind. Don't wait till the last second. Again, the German Nagoya Protocol Hub is a great place to get these resources. They have documents that walk you through the whole process and checklists so you can develop your own strategy. And use all available resources. And I don't know if the slides will be shared, but if not, then here you can do a screenshot of these resources and links that you can use. Yeah. And the other thing, all that I have discussed during this presentation mainly refers to access and utilization of tangible or physical genetic resources, but there's also quite important ongoing discussions at the CBD on how digital sequence information will be regulated in the future, or DSI. And this has been a very contentious topic for years because developing nations felt that the use of DSI was basically like loopholing around obligations that would come from the Nagoya Protocol, since there's no direct access to the physical resource. So companies or people can go into open access databases, take any sequence and use it for any commercial product, and then the provider country of that genetic resource would never get anything back. And you cannot produce DSI without the initial access to the physical resource. So after many years of deliberation at the conference of the parties meeting in December of 2022, parties agreed that a multilateral system for the use of DSI was to be established and developed, which means that the use of DSI wouldn't be treated like access to genetic resources in the sense of a bilateral agreement, but that somehow, and this is still unclear how would this be, but there will be a fund that collects all kinds of benefits from the use of DSI and then can fairly and equitably distribute it among its parties. So, and the next workshop in 2 weeks, we'll expand on this topic. So if you haven't signed up, do so, or just keep it in mind. And yeah, with that, thank you for your attention.